This is Suganti, Assistant Professor, Department of Biotechnology, Vistas, Chennai. In today's class, we are going to see in detail about prokaryotic transcription mechanism. This transcription is a very important cellular process in which the mRNA is synthesized from the DNA molecule. This mechanism is called as a transcription. So simply the RNA is synthesized from DNA using RNA polymerase enzyme. So this is one of the very first step in gene expression. In the central dogma of molecular biology, there are two different steps. One is transcription and another one is translation. This is essential for the gene expression. So the transcription is the first step of gene expression. Next, we will see some information about the RNA polymerase. RNA polymerase is the main transcription enzyme. So transcription begins when RNA polymerase binds to the promoter sequence in the DNA. Then this RNA polymerase uses one of the DNA strand as a template strand to make a new complementary RNA molecule. So before this, you must know what are the basic requirements for transcription. Ultimately, we need a template DNA and we need RNA polymerase as an enzyme. Then we need ribonucleoside triphosphate. And you must know in the DNA, one is template strand, another one is coding strand. You must know which one is template strand and another one which one is coding strand. So this is double stranded DNA. One strand is in 5' to 3' orientation, another strand is in 3' to 5' orientation. This 3' to 5' oriented strand is called as a template strand. So by using this strand only, the RNA polymerase synthesizes the RNA molecule. The opposite strand is called as a coding strand. So you must know which one is template and which one is coding. So simply you can remember like the template strand are used as a code for producing the RNA. Then you must know some detailed information about RNA polymerase. So this is the main enzyme involved in transcription mechanism. In prokaryotes, this RNA polymerase has 5 subunits. That is 2 alpha subunit, 2 beta subunit. One is beta and another one is beta prime and another one is sigma subunit. So without the sigma subunit, the RNA polymerase is called as a core enzyme. That means in this core enzyme with sigma factor is called holoenzyme. So the 2 alpha subunit and 2 beta subunit is called as a core enzyme without sigma. When sigma factor will come and attach, the enzyme is called as holoenzyme. And mainly this core enzyme only containing the catalytic activity for doing the transcription process. So this sigma factor released once it will start the transcription mechanism. So next the each unit of RNA polymerase having specific function. The alpha subunit it binds to the regulatory protein, and beta it form a phosphodiester bond, and beta prime it fixes the RNA polymerase to the DNA template, and the sigma it will recognize and binds to the promoter region of the DNA and initiate the transcription. Actually, the sigma factor is necessary only for the recognition and binding of the RNA polymerase to the promoter region of the DNA, and it's not needed for the transcription process. It's just initiate the transcription and it will fall down from the RNA polymerase during the transcription mechanism. I'm telling. Next one is there are 3 steps involved in transcription. First one is initiation, second one is elongation, then termination. So simply I have given note for each phase. Initiation means the RNA polymerase recognize the promoter and it will start the transcription. Elongation means the RNA polymerase will add the RNA nucleotides continuously and synthesizing the RNA strand. This is called as elongation phase. Then in termination phase, the RNA polymerase stops synthesis and the RNA is separated from the DNA template. Termination means the end of the transcription process. Now we will see in detail about the initiation phase and elongation phase and termination phase. So initiation is nothing but to begin the transcribing a gene. The RNA polymerase binds to the DNA of a gene at a region called promoter. So before the initiation process, you must know what is promoter. Okay, promoter is nothing but it's a DNA sequence onto which the transcription machinery binds and initiate the transcription. Here the transcription machinery is nothing but RNA polymerase. Okay, this promoter region plays a very important role in controlling the transcription of the gene. So without the promoter, there is no transcription. So this transcription factors binds to the specific nucleotides in the promoter region and assist the binding of the RNA polymerase. Okay, so you must know which one is promoter. Promoter is nothing but it's simply prior to the transcription start site. For example, here your gene is present. This region must be transcribed. Okay, so the promoter is present near to the transcribing gene. That is left to the transcribing gene. So automatically the you have to calculate the nucleotide bases in a minus. Okay, -1, -2 like it, because the transcribing gene is started from +1, +2. Okay. So in the initiation process, uh, the RNA polymerase with sigma factor will come and binds to the promoter region of the DNA. Okay, this is the promoter region here. Then after binding of the RNA polymerase to the DNA, the double-stranded DNA will open up and it will form a transcription bubble. Okay. This is called as a initiation complex. Here you have to note down the complex. Okay, here once the before the binding of the RNA polymerase, the DNA is present in a closed complex manner. After that, after the binding, it will open and it will form a bubble. So this is called as the open complex. So there are two different base sequence and the coding strand in which the RNA polymerase binds and initiate the transcription. In the promoter region, there are two different regions. Okay, one is Tata box, also called as Pigmo box in prokaryotes. It consisting of 6 nucleotide bases. It consisting the nucleotides in the order of TA. So that's why we are calling this region as a Tata box. Actually, this is located on the left side about 10 bases upstream from the starting point of the transcription. I already told, right? This is present near to the transcribing the gene in the left side. So you have to calculate in a negative -1, -2 like. So the next one is another one is -35 sequence. This is the second recognition site in the promoter region of the DNA. It contains the base sequence of TTGACA, so which is located about 35 bases upstream of the transcription starting point. Okay, and you know about closed complex and open complex, right? Okay. So the initiation is nothing but RNA polymerase with sigma factor will come and bind to the DNA. At the time, the DNA complex is called as open complex, closed complex. Then after binding of RNA polymerase into the promoter region, the double-stranded DNA will form a bubble-like structure. At the time, the structure is called as a open complex. That is simple. Okay, so initiation is very simple. Next, we'll move on to the elongation process. Here, the transcription elongation phase begins with the release of the sigma subunit from the RNA polymerase. I already told, right? For the elongation process, sigma factor will be released and the core enzyme RNA polymerase will start adding the RNA nucleotide opposite to the nucleotide which is present in the template DNA. So here you can see the synthesis of the RNA. Okay, this is elongation. Very simple. The next one is termination. The termination is a very, very important step in transcription. So RNA polymerase will keep transcribing until it gets the signal to stop. Okay, otherwise it won't get any signal, means the RNA strand will continuously synthesizing. So this process of ending the transcription is called termination. In prokaryotes, there are two classes of termination signals. One is row dependent termination, another one is row independent termination. I will explain you what is row dependent termination. So in row dependent termination, a hexameric protein called row factor, it will come and attach to the DNA strand. So automatically the RNA polymerase cannot move, and so automatically the RNA stops synthesize. So termination will occur due to the row factor. This is row dependent termination. Another one is row independent. Here the palindrome-like sequence occur at the end of the sequence. Due to this sequence, the newly synthesized RNA folds on itself and to form a hairpin loop, okay, which terminate the movement of the RNA polymerase. Here there is no row factor. The RNA itself will form a hairpin loop. Automatically, the RNA further it won't synthesis. So in this diagram, you can see the row dependent termination. Here the row factor will come and binds to the synthesizing RNA. Automatically it will terminate the RNA synthesis. In the row independent termination, the synthesizing RNA, it will automatically form a hairpin loop by itself. Okay. So it can't, the RNA polymerase, it can't move further and it won't synthesize the RNA further. So the termination is dependent upon two different things. One is row independent, another one is row dependent. Once the mRNA is synthesized after this transcription process, it must undergo some post-transcriptional modification before enter into the translation mechanism. Okay, that post-transcriptional modification we will see in detailed class in the next video. So this post-transcriptional modification means after the transcription process, some additional changes will occur in the mRNA like 5' capping, 3' poly A tail, and splicing. Okay, splicing is very important. We will see in next class about the post-transcriptional modification. Okay, thank you.